Okay. I'm not sure how to work with this thing. Hello, everyone. Thank you for still being here. And today I would like... Yes. Thank you. Today I would like to talk about benefits of using ChIP-Seq data to study evolution of regulation. I'm a part of Tuberculosis Systems Biology Consortium, and for the past 5 years, we've been comprehensively profiling tuberculosis using various platforms. In particular, we've been using our own experimental and computational pipeline to perform ChIP-Seq experiment on every single transcription factor in tuberculosis genome. So far, we've managed to successfully perform ChIP-Seq for 119 transcription factors. And it's more than 10,000 regions in total. And for the sake of today's talk, you will have to believe me that it's a very high quality data. We've performed multiple biological replicates and various validations to verify the precise location and coverage of these binding sites. For today's talk, I would like to focus on a particular subset where we've performed ChIP-Seq on 5 hub transcription factors in Mycobacterium tuberculosis and its relative Mycobacterium smegmatis. You might know that tuberculosis is a highly specialized human pathogen, while smegmatis is an environmental soil-dwelling bacteria. So while their genome conservation is approximately 65%, their lifestyle is distinctly different. So these 5 transcription factors that we've studied are very important for TB development. They're hubs, and in total, their regulons in Mycobacterium tuberculosis consist of 1,600 regions. I will start with more technical questions about conservation of these binding sites. So I'll start with a question whether sequence conservation can tell us which sites are actually experimentally validated and which are false positives of a computational motif search. Then I will ask a question whether a presence of a conserved motif in both tuberculosis and smegmatis guarantees a presence of ChIP-Seq binding site. And in general, report the statistics on ChIP-Seq conservation. And finally, I will try to persuade you that some genic binding sites are actually functional. So we've been trying to support this notion for a few years now, because once we started doing ChIP-Seq, we realized that approximately 70% of all our binding sites are genic. And we've already tried to answer this question using experimental data set in TB, where we used expression and binding data to assign function, transcriptional function, and we could assign a transcriptional function to approximately 20% of genic binding site in tuberculosis. So I will start with sequence conservation, and what we will do is we will take all tuberculosis binding site, intergenic or genic, and see how well conserved is the sequence between TB and smegmatis, and whether we can distinguish this conservation with conservation of randomly sampled motifs for these 5 transcription factors. And again, this is the actual percentage. So on the y-axis, you see the percentage of binding sites that have conserved sequence between TB and smegmatis. In dark green, you see genic binding site, and in light green, you see intergenic binding sites. And you see 2 sets of bars, which are all binding sites and high confidence, high coverage sites on the right. So first, you would notice that genic binding sites on a sequence level are more conserved than intergenic binding sites, which makes sense. However, unfortunately, if you compare the gray bars, which are the statistics for the randomized motifs, you can see that there is barely any difference between the randomized motifs and experimentally validated ones. So at least in particular between tuberculosis and smegmatis, sequence conservation is not a good parameter to distinguish false positives in a given motif search. And I won't show this data, but we used various definitions of sequence conservation, and we also involved other relatives of tuberculosis. And in general, we couldn't make sequence conservation a good predictor of experimental binding site presence. So now I would like to show you an example why sequence conservation does not equal sequence actual site presence. So here you see two tracks. The first track is ChIP-Seq in tuberculosis, and the second one is ChIP-Seq in smegmatis. And in the black box, you can see the presence of a binding site in TB and absence of a binding site in smegmatis. So this binding site is located right in the middle of an orthologous gene that is highly conserved between TB and smegmatis. And if we zoom in and look at the sequence of these binding sites, it's exactly the same. In a 100 nucleotides window, there's exactly one substitute. It's a synonymous one if someone is interested. And it's not within a motif. It's in a nonsensical position of a motif. So theoretically, the sequence should indicate that the binding site is present in both organisms. However, experimentally, we observe it in only one, in TB. So because of this, we switched from conservation of sequence to actual conservation of occupancy. Whether an actual binding site, experimental binding site, is present or absent in the genome. And obviously, when we ask this question, we need to have a way to map sites between two genomes without actually using sequence conservation. So we can use it in two ways. A less conservative one is using a target gene, what we think is being regulated in the loci. And another one is using an area conservation. We could call it a high-resolution synteny to more precisely target which binding sites we think correspond to one another. So I will give you a couple of examples so that you could get a feel for what I'm talking about. So this is an interesting example, I think, where you can see two binding sites clearly highly conserved between TB and smegmatis. And the first binding site is located in a classic divergent area near potential promoter, everything as we would expect. However, the second binding site, especially in TB, it's the first row, is located within a convergent intergenic area. So normally, if we see such a site, we would dismiss it because assigning a target to such a site is very difficult without additional information. So in this particular case, we do have that information. When we compare TB to smegmatis, we can actually see that the second binding site is actually located in a linear intergenic area in smegmatis. So what we suggest is that these binding sites are both actually regulating a single target, the one in the middle, and expression data supports that. Expression data supports that this transcription factor is inhibiting this target gene in the middle. And the mechanism of this would be inhibition through looping, which was already mentioned today earlier in the morning. And essentially the configuration of this other gene to the right doesn't matter. And when we look in the sequence, when we zoom in on that second binding site and look at the sequence, indeed its binding site is highly conserved, and the sequence starts changing downstream of it. So this example is an example in favor of highly precise mapping using both target gene and also looking at an exact configuration of a loci. However, in some cases, such as here, the right, it might be too much. So here we see two binding sites, one in TB and another in smegmatis, which we think, and expression data suggests that they're both regulating the target gene here in the middle. There's suggested inhibition. However, these binding sites are clearly located differently relative to the target gene. So in this particular case, to map these binding sites, and we are interested in knowing that this link seems to be conserved, we don't want to zoom in on the sequence. We don't want to consider loci configuration. We just want to know that it seems like it's important to keep this gene regulated by this transcription factor, and perhaps this is an example of convergent evolution. So now, keeping in mind these two ways of mapping binding sites between TB and SMIG- without actually using motif sequence conservation, we can again ask a question. What percentage of binding sites are conserved between TB and SMIG? So to the left, you see the statistics for a more conservative method of mapping, and to the right, you see the less conservative method, but both suggest the same. So first of all, intergenic binding sites now are more conserved than genic binding sites, as actually various other studies before us suggested. However, what is interesting to us is that both intergenic and genic binding sites are definitely conserved above what we would expect from random motifs. So that plays in hand with our hope to prove that genic binding sites, at least some of them, definitely have function and are not just a consequence of ChIP-Seq process. So now that we've looked into a more technical side of the story, I would like to actually look at evolution, regulon evolution in bacteria. So evolution in bacteria is actually more diverse than in eukaryotes because genomes are very dynamic. So while, yes, the transcription factor can mutate itself, however, we are working with highly conservative transcription factors that have very similar motifs and highly conservative sequence among tuberculosis relatives. So two major processes that we would like to evaluate are gene gains and losses, and also binding site rewiring, binding site gains and losses. So in eukaryote, you would normally just look at binding site gains and losses, while the genome composition is much more stable. In bacteria, it's not true. So what we did, we have some additional ChIP-Seq, in this case for an intermediate relative, which is Mycobacterium avium. So now with 3 organisms and a ChIP-Seq set for all 3 of them, we can try and actually explicitly model the rates of gene and site turnover and compare them across transcription factors. So I'm not going into details of how exactly we do this. However, it's a 3-state Markov chain where for each regulon, and we have 5 of them, and in each organism, we have 3 of them, we represent a regulon as a sequence of states, where a state for each target ortholog, each target gene, is one of these 3. So first one is an absence, as in gene is not present, and it's also obviously not regulated. Another one is gene present, however, it is not regulated. And the final one is gene is present and regulated within a particular organism and a regulon. So these are these numeric sequences, and we can estimate the parameters of these transitions for each branch of these 3. Well, so first of all, for these estimates, we kind of wanted to believe ourselves that what we are estimating looks correct. So we looked into the literature and looked at what other people saw, and thankfully we were quite glad to see that our estimates for some of the parameters match previous literature. So these 3 particular states, so multiple bars are our 5 transcription factors, and here you can see, pardon me, yes. And here you can see 3 different processes, and all these processes are the ways how regulon can stay the same between the ancestral state and the child. So either the ancestor didn't have a gene or regulation and stays the same, or the gene is maintained once the ancestor has it and the child does not anymore, or the binding site and a gene are maintained. And all 3 estimates match what is expected from the literature, and the most importantly is that binding site turnover is clearly very, binding site maintenance is very, very low, so we see a lot of binding site turnover. So on the other hand, there are ways how regulon can change, and regulon can either lose a binding site together with a gene or without it, or gain a binding site together with the gene or without it. So what was amazing to us, and that's our main result here, is that apparently evolution of TB is primarily driven by loss of binding sites together with its target genes. So this is the primary process. And then there is a small additional gain of binding sites not coupled with the gene gain. So to just wrap it up, I hope I showed you that the presence of conserved motif does not guarantee a binding site presence. Then, well, evolutionary evidence suggests that some genic binding sites are transcriptionally functional, potentially. And then for the evolution of bacterium mycetes, we see pervasive site rewiring, and also we think that the driving force behind regulon evolution in tuberculosis is loss of binding sites together coupled with gene loss. So obviously this work was, could happen because I had a lot of help from other people. So thank you for listening, and well, thank you for the community. Yes.
So how do you explain when you have a transcription factor and you have a binding site that a binding does not happen?
For example, genome accessibility. So the more prosaic answer is our capability to describe a binding motif is limited, and we are not catching certain subtle differences. However, beyond that, it would be genome accessibility. And for example, it is known in bacteria that there are histone-like proteins, such as HNS in E. coli, that protect DNA from transcription factors. And it was shown that it's one of the methods to manage transcription in bacteria. And there are known histone-like proteins in tuberculosis.
Any other questions? So the genic sites, do you see them interfering with the codon usage pattern of the rest of the gene, the general codon usage pattern in the organism?
So we see slight correlation between when a gene contains a binding site, normally it's inhibited by that transcription factor. So there is a slight correlation between inhibition, transcriptional inhibition, and presence of a binding site. Otherwise, actually, we believe that in a lot of situations, genic binding, because it's such tightly packed genome, where intergenic areas are just a small percentage of the total genome, so that in a lot of situations, a promoter of a downstream gene actually is within a coding region of a previous upstream gene. And it was previously shown that actually polymerase can essentially go through quite a few inhibitors without stalling. So polymerase has a much higher affinity to DNA than transcription factors. So it can just roll through.
Questions?
Thank you.